Hello. So, today we will continue those nickel enzymes, and today we will see first that some of the model compounds in their involvement in say bimetallic catalysis. So, is a very well known term nowadays, that what do we mean, that if you have two metal centers, and basically while we study all these informations, that any particular motif, whether it is a biological motif or a synthetically prepared molecule, the two metal centers are present. So, whatever informations we are getting, so nature is giving that inspiration to know that particular thing. And one important aspect for that catalysis would be that how you place two metal centers. It can be a 2 iron center or it can be a 2 nickel center, because today we will see some molecules, synthetic molecules, where 2 nickel centers are present. So, this basically involves the ligand design, which is the most important part of this particular study. That ligand design, that means what particular type of ligand you can have, and you get basically two distinct pockets. In biological system also the protein gives that particular pocket. So, this is basically your pocket 1 and another is your pocket 2. And how you can fix this particular distance, because while you go for this particular type of catalysis, and always we basically think that one particular site, that means the metal 1 and metal 2. So, this particular site is basically holding your substrate. If we are talking simple urease activity, so urea should go and bind that particular site, and the other site which will be your reagent, that means the nucleophile. It can be your water molecule or it can be your hydroxide anion, and then we get something that this particular thing, that means the reagent and substrate will react, giving you your product. So, that is the idea, that why we take these two metal centers in a particular system. So, these two metal centers, that means their coordination geometry will play an important role, that what type of ligand you should take and what are the connectivities. So, if we can have one single connectivity, that means we put X over here. So, one pocket which is elaborated around X and another pocket which is elaborated on the other side, and if we get M1 here and M2 here, and this particular group is utilized for bridging. So, this is basically a single point attachment. So, singly bridged binuclear system. So, X is there. So, singly bridged attachment if you have, then you can tell something related to your distance. So, it is say D1. Then in the next case, that means you can have some time, you can bring the second group, that also can go for the bridging. And when you have a situation like this, in X and Y both are involved for bridging, you can decrease the corresponding distance between M1 and M2. And remember that whenever we bind this thing, always you should have some vacant positions. So, designing this thing, we always require some positions which should be vacant. So, these two vacant site can be utilized for substrate binding and the second site for reagent binding. And if X is there and X is part of the ligand, during the ligand design, if you think that X should be there and based on that X you get the corresponding binuclear compound, and this one which is coming from outside, if this X we call it as a endogenous one which is coming from the ligand system, then Y is exogenous. That means some small group, like sometime we will find a labile group is also fine, water molecule. So, it can be water breached. Why it is water breached? Because X is there and some other donor points, if it is available for binding M1 and M2, you have some other positions available which can either be occupied by monodentate water molecule, but if this M1 and M2 are close enough, you get a bridging water molecule or a hydroxide group or a oxido function. And depending upon that, that means depending upon their protonation level and the pH of the medium, you can move from here to here or here to water molecule. And interestingly, when you make this, and if it goes for some kind of loose binding on M1 and M2, we will find that if this water bridge cleaves, you get two water molecules which are bound to the system in monodentate fashion. So, if you get ultimately this arrangement, this is also suitable for your substrate binding and reagent binding, because these water molecule can be substituted by your incoming substrate molecule, and this water molecule, if water is not required, something else is required for attacking your substrate as reagent or as some useful group which will attack your substrate. So, these two water molecules will be there. So, what is this X? How this X can be modified or how this X can change the different distances. So, we will see this in some model compound, that means synthetic compounds, what we can make in the laboratory. So, what we have seen that the bridging coordination mode of urea, when we are talking about urease molecule. So, this is one such motif, do not worry about the complex structure for that. So, if you have this N1 and N2, what we need? We need one pocket for binding the first nickel and another pocket for binding the second nickel. And this particular one, so this is the endogenous bridging unit which is based on pyrazole. So, pyrazole group is utilized for holding this nickel and that nickel through its NN bridging mode. So, NN bridging mode on the pyrazole, and this pyrazole group you have other tridentate groups you can make on the other side. So, these three, this nitrogen and this nitrogen can give you a facial capping on the other side of the nickel, and this particular nitrogen is giving you the fourth coordination. So, you have 3 from this capping part and 3 from the other side. So, you have 4 coordinated nickel on this side and another 4 coordinated nickel on the other side. So, these two nickel centers, when they are coordinatively saturated, you get a pentacoordinated species, and you have something which is nothing but, since you have some vacancies, some positions, it is sometime it only bridged by hydroxide group or is the dimer of hydroxide and water molecule. So, water molecule, so it is a basically water hydroxo cluster is there utilizing for your binding the nickel group. So, this bridging coordination mode of urea and carbamide, that people have identified in this paper, that the groups which are responsible for binding a dinickel site, because model studies gives us all these informations related to the urease activity. So, we get a bimetallic complex, and using a reaction where you have the trimethyl silyl urea, this R2 R prime is trimethyl silyl function. So, basically is that you get the corresponding urea binding in this form and you have NH in the deprotonated form. So, it is not the pure substrate as urea, but the binding mode people have tried to identify it, that this can be your substrate molecule which is binding. So, you have the nice cavity, that means the nice vacant positions where you can displace this water and hydroxide groups by your substituted urea molecule. So, which therefore can be a relevant part for the active site of metal enzyme in urease molecule. So, this is the typical reaction condition where you get, and you will find that you have something that you get the corresponding species, that the nickel-nickel distance is important. So, these molecules people have prepared it and then structurally characterized. So, this particular nickel-nickel distance will tell you whether this nickel-nickel distance for compound 1 is suitable for bridging hydroxo function or a hydroxide water group for longer distance of nickel-nickel, because you have here, when it is replaced by these groups, you have a bridging unit which is a 3-atom bridging unit like that of our acetate function. So, when acetate group bridges 2 nickel centers through oxygen, carbon, oxygen like your copper acetate, you have reasonably bigger distance compared to a single atom bridging, compared to bridging by a hydroxide function, a methoxide function or single oxygen of acetate group. So, this distance is reasonably big enough, because we get in the second reaction where ammonium carbamate is used. So, directly the hydrolyzed product of this urea hydrolyzed product which is forming there, we just utilize, they have utilized it, because whether your urea that ammonium carbamate is going there and is binding these 2 nickel sites. So, they are basically prepared and structurally characterized, and in one case, that means this bridging unit you can have large number of this, what I am just told you, that you have this exogenous function, an exogenous function when you have the nearby second metal, that means this bimetallic system or the bimetallic complex what you can have, that is not that you have 2 such groups, that means 2 urea molecules or 2 water molecules are coordinating to each nickel site in monodentate fashion. So, your distance will always attract, because this particular site will be remain vacant if you have water coordination and only that urea molecule is binding the first nickel in monodentate fashion. That is not happening there. And this in the second case also. So, this thing, this particular core, you look at the core basically. So, this if you consider these 2 nitrogen, and this is the 3-atom bridging unit, oxygen, carbon, oxygen. So, 3 plus 2, 5 plus 2 from the pyrazole unit. So, it is a 7-membered ring. So, this 7-membered ring obviously will tell you that immediately by looking at the bridging units which are utilized for bridging, you can immediately say that this nickel-nickel distance is bigger enough compared to some bridging what we will see in some few other examples, that this can be your phenol based also, because in earlier systems and other systems we know that this can be your phenol. That means, the phenol oxygen can be utilized to hold 2 nickel sites. So, when you have one single atom here, another single atom here and 2 nickel, so that basically N2O2 core, that is also possible to have, where you have the shortest nickel-nickel distance. So, these things, if you get the corresponding structure and all these and the positioning of these 2 nickel centers, you can characterize something related to your magnetic interactions also, that people do for the magnetic interactions where these nickel 2 sites are present. So, you have the deprotonated urea and carbamate which nicely binds to 2 nickel sites. So, this particular structure basically in the acetone solution of 1 urea and bis trimethyl silyl urea. So, is a substituted version of urea, and the monitoring can also be possible. It is not that you have the structure and you determine the structure, then only you will be able to tell whether you get the corresponding substitution by urea or the carbamate group. But if you simply notice the corresponding change in the UV visible spectrum, that means there is a slight change in the corresponding color of the green compound, because this nickel you have the strong DD band for the nickel 2, because both the 2 centers we are not talking anything related to the nickel in other oxidation state, that means nickel in +1 or nickel in +3. Here basically we will get nickel in +2. So, the DD transition will be predominating and you have the green compound of this, and when it is reacting with the incoming group, that means the exogenous function, any exogenous function. So, any exogenous group will coordinate. So, you get something, that means your corresponding ordering for the D levels are modifying and there is a slight change in all these lambda max values. So, if you have a spectrophotometer and if you monitor the corresponding DD transition bands, you will see that there is little bit change for that due to this sort of coordination. So, the positioning of nickel, two nickel sites with respect to this pyrazole unit, this is your pyrazole unit, and the corresponding binding of this group can also tell us something related to that of your corresponding sites. That means, when we have this M1 and M2, we get these substrate sites and the reagent binding sites. These are your, basically we call them as active sites. So, what is there? That means, whether your active site is vacant or they are binding to water molecule or they are utilizing some other donor groups, because the availability of these active sites are very much important for their reactivity of the enzymatic site. So, the reactions for those active sites is important. So, if these are not available, that means the vacant sites are not available, and if they are water bound or the bridge groups, if they are occupied by some strong groups, say N3 minus or thiocyanate or selenocyanate, etc. So, addition of these groups, like what we just have seen, that the binding of urea and the binding of the carbamate groups changes your corresponding pattern in UV visible spectrum. So, the corresponding lambda max values for the binding of these groups individually or separately, that can also be studied nicely. And if they are bound strongly, because we are talking in terms of the binding of urea and its transformed form as carbamate, but when azide is binding to these sites, either the substrate site or the reagent site, and if they make some strong nickel nitrogen bond, so nickel and then azide. So, if they are making some strong bonds to that, and basically this particular catalytic site will lose their catalytic activity, because you cannot remove the azide from the system. So, that particular activity, that means, if we are thinking of some activity related to urease activity, so that activity will be inhibited. So, inhibition study, that catalytic inhibition study can also be done by looking at some of these exogenous useful groups, that exogenous groups which are coming there and binding to the nickel site, by simply looking at your corresponding lambda max values. So, that gives us not only the substrate binding or its transformed product, but also some kind of inhibition, if it is possible to achieve there at the active site, that can also be studied. So, these other two examples is basically the same molecule, where you can have these hydroxide groups. So, in this particular case, where you see that a bigger unit was present, where you have this water molecule, this water is there and you have the hydroxide function. So, this particular unit, so basically what is happening there, that individually this particular pyrazole motif is so important, but you have the corresponding two bonds, because whatever coordination geometry you are achieving on the nickel side is important. If it is pentacoordinated one, you should think of what type of pentacoordinated geometry you are getting around nickel 1 and around nickel 2. Because if it is a simple pentacoordinated one, you can have a geometry of square pyramidal one or a trigonal bipyramidal one. And if you have a square pyramidal and bipyramidal geometry, then which particular bond is available. If you have all the positions are there, so which particular bond is available for binding this loose water molecule, that you should think of. So, whether it is typically in this direction or it will be in some angular direction. So, basically just what we have seen, that you can have two nickel sites, and the possibility is also there that two of these sites can be occupied by two water molecules. Here this water molecule has undergone deprotonation only. So, this is your hydroxide of bridge function and this is your water molecule. And you see, if you bring any substrate molecule like that of your urea, that urea will immediately replace first what? That water molecule. So, you will have a urea bound water molecule there, and in the second nickel site is having some hydroxide group. So, this metal bound hydroxide group will then immediately attack your urea molecule. So, urea carbon will be attacked by your hydroxide function. So, the flexibility of this particular backbone, because the protein structure what we can have in the nature, in the biology what we get, the protein structure is so much flexible that the flexibility of this system will say something, that you can have sometime you will find that depending upon in one particular form, the particular enzyme can have different forms depending upon some protonation level. That means one particular site is protonated and another site is deprotonated. You have the corresponding interaction, that means the bond is typically parallel. These two bonds, that means it is monodentate groups which is attaching nickel 1 and nickel 2 are almost parallel. Then you have one parallel water coordination to this site and another parallel water coordination to that site. In that case, you can have the individual water molecules attaching to this site and attaching to that site will not form any kind of this hydrogen bonding interactions. When they are angular, because basically we find these O distance. So, when they are in hydrogen bonding distance, then only it forms some hydrogen bonding interaction, and that is basically governed by the typical endogenous backbone of the ligand, that means the pyrazole backbone. If the pyrazole backbone is controlling, because the most favored geometry for that is that the one side, this other side you have a pyrazole unit, that means you have NN bridging. From this side you have NN bridging. So, what you expect? You expect from the other side you will have also a NN type of O type of bridging, that such that you can have a symmetrical 6-membered ring. 6-membered core ring is formed involving two nickel sites. Okay. So, what we will now see that how you change this particular pyrazole backbone. That means this bridging unit is so important, and while talking this all these model compounds, you have to have this particular primary coordination site, which is very important. Otherwise, you cannot bind two nickel sites like that of your protein structure, because the protein have a very huge system and you have the already made that cavity and other cavity, and those two cavities basically bringing two nickel sites close to each other. But here you need the basic support from this parent unit, either it is a phenol unit or a pyrazole unit, and you can have at least one coordination site out of this as this nitrogen and one site as that nitrogen. Otherwise, if you have a simple pyrazole unit like pyrazole, imidazole and all these nitrogen bearing heterocycles, if you do not force, that means if you do not have such type of chelating arm like this is the NN chelating arm of this pyrazole elaborated ligand, and this is another NN chelating arm on the other side. So, if you do not have this chelate arm, it will not hold the first nickel, similarly it will not hold the second nickel by the simple pyrazole unit. So, if some supporting system is there, then only pyrazole can bind to these two nickel sites as a bridging unit. Otherwise, pyrazole can be happy and can stay as a monodentate unit to these particular sites. So, this is the second example where you just take the elaboration, that means how large number of any bimetallic sites. This is some I am citing some of these examples where you can have a particular unit. That means, whenever we talk that any bimetallic catalysis or bimetallic units are required, that you have to bring 2 metal centers, not more than that, at least 2. So, the very basic minimum number of metal centers you require for this sort of catalysis is a very simple thing, is like pyrazole. You now take the phenol unit. So, the same thing, the phenol unit, and from these 2 ortho positions you elaborate this group, and again you bring 1 nitrogen here. So, it is elaborated through this nitrogen and it is elaborated through this nitrogen also. So, including this nitrogen and this phenol ring, you have the corresponding size of this ring. So, you have the 6-membered ring. So, this is a 6-membered ring, and on the other side also you have a 6-membered ring. So, that 6-membered ring will be there. So, that is the give you the corresponding stability rendered on the nickel 1 and nickel 2. So, it is very simple to make it also, because if you have at least 2 substitutions on these positions on the phenol units at the 2 position and the 6 positions. So, this is the whole name of this ligand based on phenol, and you have the ligand and very simple salt, that any salt from your periodic table you can have. So, it is one example is that for the dinickel compound in 2 plus 2 oxidation state, because we are talking about phenol, when it is based on phenol, you do not expect that you will be able to stabilize nickel in the lower oxidation state. So, already you have the charge, and phenol is well known to stabilize the nickel in the most normal oxidation state of nickel, say plus 2. So, you are providing something, that means you are giving nickel perchlorate over here. So, is the most suitable salt, because the perchlorate groups you can remove very easily. Then give this ligand. So, not the nitrogen, but you elaborate further, because these are the 2 other pyridine units. So, these 2 pyridine units. So, if you consider on the left hand side this nitrogen and this nitrogen. So, look at the molecular structure, how you read. It is not that you should be a very expert coordination chemist. You look at the nitrogens available which are binding to the nickel side, because the protein substrates are coming like this. The histidine side chain, all the amino acid side chains are coming like to bind the nickel side. So, we have these 3 nitrogens. So, how we make this elaboration? This is basically a part of the pyridine ring. This is also a part of the pyridine ring, and this nitrogen is the aliphatic nitrogen which is directly attached to your phenol unit. So, what you can have? So, if you do not go for all this complex system, that individually we make basically this tridentate ligand all the time you can make. So, if you have this pyridine based system, so 2 pyridine groups you can have. So, if you take any pyridine where you have in ortho position you have the amine function. So, in any pyridine ring you have in the ortho positions methylamine function, and you take the second pyridine ring as your pyridine 2 aldehyde, and you get the corresponding CYP base. So, a simple CYP base you will get, and that CYP base will be a tridentate CYP base. So, this nitrogen, the second nitrogen and the third nitrogen. So, if you have the tridentate ligand, and if you reduce the tridentate ligand, and the next step of this making this molecule is that how you put the tridentate ligand to the phenol unit. So, simple course of reaction is a simple Mannich reaction is possible, or if you can have CH2Br substitution on these 2 positions. So, that immediately react with the reduced form of this, that means you have the NH function and you attach this part to this group. So, the similar arrangement for the other side, but you have something, because this particular example will give you not only when you force this particular system, because you react with nickel perchlorate, the ligand and sodium acetate. Why we are not using something? Because sometimes the solubility and the crystallization is so important, because you cannot use nickel acetate for that reaction. So, initially we do the reaction with perchlorate, then externally you are adding sodium acetate, because initially what you are looking for, that these positions what are occupied by these 2 acetate groups, these are your acetate groups. So, initially when you react with perchlorate, nickel perchlorate, those will be occupied loosely either by the perchlorate anions or by water molecules. So, these will be the loosely attached groups over there. And in this particular case you have the pocket. So, the nickel center in both the cases it is similar, but instead of getting this particular part, so on the left hand side, so this structure is on the left hand side, you have a tridentate part from this nitrogen and that nitrogen, but on the right hand side you see is a different one. So, we make something. So, this particular binucleating ligand is some example where these two pockets are different, it is not a symmetrical binucleating ligand. So, protein structures are also sometimes like this, that means your available histidine groups and all these groups are not matching in number for the left side of the cavity than your right side. So, if you take something, that means you on the right hand side you do not allow a tridentate ligand to bind, but also give you a bidentate one. So, you will have only 2 nitrogens available from the right hand side. So, one site is available which is nicely occupied by simple water molecule. So, that is why this water in the formula it is there, this water molecule. This water molecule is there on the right hand side. So, what basically getting from here, that is the same dinickel compound, and this can also be considered as a model complex for urease activity, because these acetate functions, the next step what you will be able to do is that you remove these acetate groups. So, if you just loosely go for some bisalt or some weak acid, you protonate these acetate function and acetate functions will go simply, leaving behind the corresponding acid as again perchlorate, and you react with the urease molecule, urea molecule and the water or the hydroxide function, those will be occupied by these groups. But other interesting aspect, this is the chance that I can tell you here immediately, because we will not study all these things, because the much more complex system in the enzymatic function is a heterobinuclear system. So, some hydrolytic enzymes like purple acid phosphatase type of thing, or what we have seen in case of superoxide dismutases, that you have a center which is superoxide dismutases are copper based system, and one particular site is occupied by zinc also. So, that is a copper zinc system. And if you go for purple acid phosphatases, which is a iron zinc system. So, you have a pocket where you will find from this molecule, basically if you do not prepare any such molecule, but if you look at the structure nicely for this molecule, immediately you can think that okay, I can replace this particular point by changing the metal salt for the reaction as iron. Iron will go inside nicely in presence of substituting this particular nickel site. So, nickel is not there. So, iron will sit nicely over here, and this particular site is also suitable for binding zinc, and when zinc is binding over there, the most preferred coordination number for zinc is 4 and 5. So, it is this water molecule will not go there, and it will be closely your square pyramidal geometry. So, if zinc is sitting over here, you get a system where you have a square pyramidal geometry for the zinc and you have the octahedral geometry for the iron. So, that sort of thing. So, you generate some asymmetry within the binucleating ligand, and you will be able to make any compound, like you can put iron on the left pocket and zinc on the right pocket, and you have the bridging unit, because you can modify these groups, because when we talk for the simple urease molecule, and that urease molecule we are talking about some hydrolytic reaction. So, when we talk about some ester hydrolysis, say phosphate ester hydrolysis, so those group of molecules are the similar type of molecule, those are phosphatases. So, one such example I am just telling, we call it as a PAP, the purple acid phosphatase. So, that purple acid phosphatase is also similar, where one site like that of superoxide dismutase is basically zinc is present, and if zinc is involved there and iron is involved there, and we are not going for any kind of redox reaction, is a hydrolytic reaction. So, zinc is fine. So, any water bound or hydroxide bind group on zinc is useful for hydrolyzing your substrate molecule, which is your phosphate ester. So, phosphate ester will be hydrolyzed over there and will remain like your acetate function, they are corresponding phosphoric acid, substituted phosphoric acid will remain there and which will be bound to your metal center. Okay. So, this gives us some idea that how you go for some model compound for dinickel system, model compound for heterobimetallic system like copper zinc or iron zinc system. So, one such example, simple example that we can go for this type of thing, that when we are handling these molecules, we are talking, that means we are handling something related to our nickel salt. So, we just go for some group where you have the nickel nitrate as the salt, and this is another example of a binucleating ligand, and it is a very simple ligand you can make with a long chain amine ligand, which is nothing but is a well known one, is a triethylene tetraamine. So, if you have a triethylene tetraamine, these 4 nitrogens are coming from triethylene tetraamine backbone. So, very simple way, if you react triethylene tetraamine with salicylaldehyde. So, on two ends you have the salicylaldehyde as the sheaf base, but here because of the availability of the two adjacent NH functions, you can make some imidazolidine ring also. So, once you are able to make some imidazolidine ring on the adjacent nitrogen, you basically compartmentalize this hexadentate ligand into 2 form. So, this is basically your backbone, that means like that of your pyrazole unit or phenol unit. So, this phenol unit which is attached to your imidazolidine ring can go for bridging to nickel site. So, you have some group, but what I am giving this example, because your ligand is not very much strong, because in situ you can make, when you add, is a very simple reaction even anybody can make in the laboratory, that you have to take the triethylene tetraamine and salicylaldehyde. So, that ligand if you can have, and that gives large number of reactivity pattern for that when you react. So, one particular reaction condition. So, this gives us some idea that why these reaction conditions are important. How you get that? You do not know whether you will get a compound like 3 or a compound like 2 or a compound like 1. So, in one sort, one example is giving you that you can get that when you react it directly with this particular imidazolidine based binucleating ligand. This is the ligand which is written as H3L. You have 3 protons on these 3 phenol groups, one this, the second and the third. So, when it is reacting with this, you are getting some mononuclear compound. So, that means you must have a very good backbone. The stability of the backbone which we are talking about, say your pyrazole backbone or your phenol backbone, the stability of the backbone is important, but in this particular case you have the substituted imidazolidine ring, and that substituted imidazolidine ring is not so stable, is different from your imidazole ring, is imidazolidine, which is a reduced form of imidazole and which we make from diamine and aldehyde. So, diamine and aldehyde immediately gives us imidazolidine ring. So, that imidazolidine ring is also very much susceptible for hydrolysis reaction. So, in one sort what we are getting, that in urease reactivity, the nickel center which is bound to some water molecule or some hydroxide group is responsible for the hydrolysis of urea, but in this particular case if your ligand is not so stable and nickel is present and you are doing all these reactions are in aqueous methanolic medium, we are not maintaining any dry condition or some inert atmosphere. So, methanol is there, so water molecule is also there. So, water is interacting. So, because these 6 water molecules for the nickel nitrate, starting nickel nitrates, you have plenty of water molecules already present. So, those water molecules already there. So, nickel bound water molecules, remember it. So, you have nickel bound water molecule, and when we are allowing it to react with your ligand, so your nickel bound water molecule is your reagent now, which is your catalyst, is a reagent for hydrolyzing your imidazolidine backbone. So, similar to that of your urease activity, so it is hydrolyzing the imidazolidine backbone, leading behind, that means this particular phenol unit, that means the salicylaldehyde group, simply when you go for this reaction, your salicylaldehyde group will go away. So, it is just simply removing from there. So, originally if you have this ligand, because you see this ligand, so here you have the intact this particular part, you have this intact part. This on the left hand side what you have? You have 2 nitrogen and 2 oxygen, because this oxygen is your phenol bridging unit. So, you have the N2O4 pocket on the left, and on the right hand side also you have N2O4 pocket. But when this particular group is going away, that means removing one salicylaldehyde molecule from the system, you are ending with a simple hexadentate ligand. So, this is a typical hexadentate N4O2 ligand, and nickel will be nicely coordinating to all 6 donor groups and giving a corresponding complex. But the thing is that the situation is depending upon the condition of that, and you are not adding some strong base, because in most of these cases when we talk about the coordination of phenol to any metal salt or any phenol substrate you are using, and in presence of the metal salt, if you do not put appropriate base, say simple triethylamine. So, giving some appropriate base is very important, otherwise you do not get the corresponding deprotonation of the phenol unit. Here we have directly reacting this, because basically we are refluxing in methanol for 1 hour with this ligand, giving you some compound. Already you have the nitrate groups present. You see this compound which has been isolated as good crystal, and you have crystallized it in the single crystals also from the parent solution. So, this compound is crystallizing as a monocationic compound, not a neutral compound, because you have these 2 positions already. If they go for deprotonation, you get a corresponding neutral compound. But this particular OH, you see like that nickel has some affinity for binding water molecule, it has some affinity for binding hydroxide function. Similarly, in this particular molecule, a simple mononuclear nickel compound, but it gives, in this particular case you have the deprotonated phenol oxygen, you have a short nickel oxygen bond, but this nickel oxygen bond is long enough because you have coordinated phenol group. So, this particular nickel will also gives us some information that this nickel is coming out from the medium, where you have both the 2 phenol units in different protonation level, one is in the deprotonated form, another in the protonated form. So, what basically we see that if you have the nickel nitrate, and if you use this particular type of ligand, which is a very fragile ligand, we can call is a fragile ligand, because it is getting hydrolyzed, it is not so stable, that means your backbone is not so stable, it is not retaining nicely. So, backbone can go away for any kind of nickel based hydrolytic reaction. So, what do you think of that if you use in a reverse way, that when we are talking about in all these biological system, all this enzymatic nickel system, that if you have this particular phenol based dialdehyde, 2,6 diformyl phenol, 4 position is also substituted by methyl group. So, this particular unit, what we just talking about the generation of this ligand or any ligand in protein chain also, if they are elaborated based on tyrosine residue. So, tyrosine amino acid residues, why this tyrosine is so important in all these sort of interactions with the metal salt, why these tyrosine residues in some point it is giving rise to the free radicals for the galactose oxidase activity and all this. So, binding of this phenol unit is important, because if you do not have these groups, that means this aldehyde functions, if you do not have, it is very difficult to go for corresponding binding of this phenol unit. Phenol can bind to any single metal salt in monodentate fashion only. But if you have some groups like that simple aldehyde function, so you get this, that means if you have on the other one side, that means this particular unit on the right hand side, this is your salicylaldehyde. So, you see that we are not doing all these reactions, because the well known corresponding bidentate OO type of ligand is, we all know, that is acetylacetone, acetylacetone. So, salicylaldehyde is also behaving in same fashion, that simple salicylaldehyde can be a very good bidentate ligand, we will come there actually. So, right now you see that elaboration on the both side of the phenol unit, if in the form of that phenyl ring, or you bring these 2 groups from the protein chain. If you are able to bring these 2 protein chain groups from the protein chain from histidine residue or any other residue. So, you can put one metal here and you can put another metal. So, that basically gives us. So, whatever you react with the nickel nitrate with that of your this particular dialdehyde, you get some particular motif like this. So, in this particular environment, that means the nickel nitrate salt. So, nickel nitrate salt is giving you a particular unit which is binucleating. So, very simple thing, that means you have any simple such ligand, anything you can think of, anything you can make and you can allow it to react. So, always you think of that whatever this type of unit you allow it to react with any simple metal salt or nickel nitrate, nickel perchlorate or anything, you always think of this motif. That means, you are forcing your phenol unit to bridge these two nickel sites. So, then you have in your hand this same ligand H3L. So, when you allow it to react with HCl, because this particular unit very readily you can isolate it, and when this exogenous group is present, you do not find any kind of hydrolysis. So, if the condition is same in presence of this group, the same ligand in methanol reflux for 1 hour. So, condition is same, but you are getting two different compound 1 and 3. So, the presence of your supporting unit, that means you can consider this now as your exogenous group. So, exogenous bridging unit. So, anything like your azide function, your thiocyanate or hydroxide function, you can consider as a exogenous unit, because this particular part is not part of your ligand unit. Your ligand unit is this one above. So, this particular, so it is basically a motif. So, in solution what we are getting in solution, you basically you can have some motif, because you can have all these loosely bound nitrate groups, water molecules and all, these are all loosely bound thing. So, on this dialdehyde platform, you can have a dinuclear entity, the dinickel complex you can make in the solution, and above that particular system, because if you think of some catalyst environment, you can go for some catalytic reaction. So, if you are thinking or if you can talking about something related to a nickel catalysis, so you take the nickel salt, you put something, some support mechanism, that where you have some exogenous ligand which can support these two nickel sites, and you get this. So, this particular thing is forming, that is why you are able to stabilize this particular unit and is a symmetrical one. So, in our previous example where we have seen that you can have an unsymmetrical binucleating ligand, but this ligand is a symmetrical one. So, both the two pockets are same. But if you change and what you are reacting, your exogenous ligand is also symmetrical, which is forming phenol aldehyde group and phenol carbonyl function for this nickel as well as this nickel. But if you same thing, that means elaboration of this ligand, that if you get this one with dialdehyde, you should get it with monoaldehyde like salicylaldehyde. So, when we react with salicylaldehyde, the starting compound what is forming in the solution, and you get this particular one. And in this particular case, you see we are forcing, because this particular sort of motif is not easily available in the system, where your salicylaldehyde is functioning as a bridging unit. And again, whatever we are discussing so far about the positioning of these two nickel sites are important, and these two positioning of these nickel sites is important. Otherwise, you can have this particular unit if these nickel 2 are little bit different, if the distance is beyond 3.8 or 3.9, you get this nickel is bound to 2 water molecule, this nickel, and on the right hand side you have only bidentate salicylaldehyde, what you expect. But since the nickel distance is short, you start some interaction with this phenol unit, and this phenol group is utilizing for bridging between these two nickel and one such group. So, your exogenous group is not that all the different sorts of exogenous group can bind a dinickel motif symmetrically. So, it is a asymmetric binding. So, exogenous group is asymmetrically bound to these two nickel sites, and if you think of something related to that particular type what we are getting for dinickel system, that this can be your substrate. We are talking about something binding of urea, but instead of urea what you can have in your hand is now a carbonyl function which is coming from the salicylaldehyde, and this carbonyl group is nicely coordinating to your nickel site, and the second site is having some your nucleophile, that means the water molecule. So, this is the situation where you can have the reagent in your hand, that means the water is bound to one nickel side and your substrate or carbonyl function, which is similar to that of your urea coordination, is attached to your second center. So, from a simple ligand system you can have 3 different types of molecules which can give rise some information, some understanding about your urease activity. And if you go for the further reactions, that means when you talk about something, that means if you have a ligand system and that ligand system is in your hand and that ligand system is utilized for a nice dinickel system. So, this is your dinickel system and salicylaldehyde is utilized for bridging these 2 nickel sides. But this particular unit, when we talk in terms of the corresponding reactivity pattern, so these complexes, these binuclear nickel complexes are very much reactive in. So, the reactivity pattern, these are the 2 such examples, that if you go for what we are talking just now, that if you are allowing it to react with sodium azide. So, this compound 2, whether you can substitute these corresponding positions, that means whether your water molecule, this particular group was attached to your water molecule, is occupying by your azide function, that means what we are talking in terms of the corresponding poisoning or inhibition of the catalytic site. So, that we should know how good they are for reacting some exogenous group like azide or thiocyanate or any other hydroxide function. So, when you allow it to react with this azide function, so they are substituting this water molecule and this salicylaldehyde function is still remains, it is not going away, but interestingly we get some different architecture. So, sometimes in solution chemistry and in the biological system, when we study all these biological sites, these particular groups are very difficult to understand, that when you have this particular unit. So, the reactivity pattern for this. So, if you have, so when you are utilizing this sodium azide, it is not that in biological system you are providing both together, that means you are providing the cation and you are providing the anion together. So, when we are providing in a synthetic molecule, so that is why the synthetic chemistry is so important, and you get something, that means how your anion is entering into the system and how your cation is entering into the system. And if you, because everything is now in front of you and you know the entire structure, only thing that you have to justify the reaction, we are justifying the reaction, why it is happening. If something is given to you, the ligand is given to you, should be able to tell that why it is binding in this fashion and why it is simply transforming for a very simple reaction with that of your sodium azide. So, before talking in terms of this, you just simply go for another sodium salt, which is your sodium perchlorate. So, this compound, you see the reactivity pattern, because if you can recall the thing that this particular compound is definitely, we have identified this compound is not a neutral compound, because all the time what type of compound you are getting, whether a cationic compound or neutral compound, because when we talk in terms of interaction with some other biological stuff, say simple interaction with the DNA molecule, which is anionic. So, if you have some cationic unit and that cationic unit nicely goes into the DNA site, so the cation will have a typical interaction for that, but those are much more complex interaction, that is very difficult to identify or understand sometime. But before that, if you have a corresponding cationic compound in your hand, so definitely we have not able to understand that thing, but initially when they are reacting, because if you can monitor the corresponding reaction kinetic, initially all the 4 nitrogens, the nitrogens are all neutral in nature. So, all the 4 nitrogens are available to bind your nickel site. Then your phenol groups, because they are all protonated, so they will have some weak reactivity pattern for the nickel site. So, they are reacting with your nickel site. So, this particular system is there, that means if you provide, that means if you are able, because this particular thing, if you just simply go for potentiometric acid base titration, based on that you will be able to deprotonate this hydrogen and you get the neutral compound. So, deprotonation will be possible when you have the phenol bound to the nickel side, because you have this particular OH is much more acidic compared to your free phenol. Your pKa value for this OH is less compared to your typical phenol what we know for the isolated molecule. So, this is your neutral compound, and that neutral compound, when you allow it to react with say sodium perchlorate. So, instead of going for some protonation, because you have not provided any proton source to the system, you have provided instead a cation, and this particular cation. So, this thing, that means basically this binuclear nickel side, because all the time when we talk in terms of the biological system, you have large number of these cationic species. When we talk about the corresponding sodium pump or the potassium pump in the biological system in one cell, so you have plenty of sodium side. But when we talk in terms of the corresponding nickel side and which is bound to the phenol unit, so some groups are still available. So, you see in this particular synthetic compound what we can characterize it, and we can solve the structure for its typical identification. So, ultimate identification for excess structure determination is possible. So, if you have some already phenol groups which is bound to the nickel side is available there, this sodium is go for coordination like that of your typical thioether binding. So, crown ethers are well known, you all know. So, the crown ether type of coordination we all know. So, if you can have a 3 oxygen atoms in a cyclic crown ether type of arrangement, it nicely binds your sodium or the potassium ion. So, that sort of situation if you are able to give with that of your dinickel compound, because you have 1, 2, 3, these 3 oxygens, if you see the corresponding structure for these, you will find they are positions nicely over there. That means, these 2, 3 oxygen atoms are closely disposed like that of your corresponding cyclic crown ether. 3 oxygen crown ether we know like that of your triisocyclonone type of thing, the trioxacyclonone. So, trioxacyclonone if you can have. So, if you available 3 oxygen which is well known to bind your sodium site, but in this particular case this oxygen is already utilized for its bridging groups to these 2 nickel. So, this oxygen is not available for binding your sodium site, and these 2 terminal oxygen groups only can bind your sodium, and sodium is also attracting one water molecule. So, your neutral compound, you are forcing it to again a cationic compound, but not providing any protonation on the phenol groups, but putting some sodium into the system. So, what in this fashion, this is another avenue that gives us some idea that how you immediately convert a corresponding neutral compound, a neutral binuclear or a dinickel compound, you can immediately convert it to a corresponding cationic compound if you put sodium within it and you have the corresponding typical anion outside that. So, if you get that particular thing, and if you have already this thing, that means what is happening there, instead of perchlorate, if you have azide function. So, in this perchlorate compound you have water bound to it. So, this loosely bound water will be immediately replaced by your azide function. So, depending upon your corresponding position and all these things, and we all the time you can think of that azide coordination can replace your salicylaldehyde molecule, but it is not that. So, it is a very delicate molecule, and so many things are there, because the whole dinuclear compound is already present over there, and that dinuclear compound you do not find any ligand exchange reaction. Your salicylaldehyde group is not moving away, salicylaldehyde group is, because it is bridged come chelation is there. So, it is strongly holding the molecule, and azide function replacing this water molecule, and you have a different type of thing. That means, this azide end, the other nitrogen is close to the sodium. So, sodium is now involved for bridging. So, is a basically coordination polymer, 1D chain is forming, and that chain is forming is a very unlike unit, that when you supply both the sodium and azide, azide is coming and occupying the sodium site, and already through this reaction we have seen that sodium can bound. So, this is binding through ligand oxygen end only. So, basically we are decorating the molecule by putting sodium on the 2 oxygen. So, these are basically binding on the oxygen. So, because this sodium is again a metal center, cannot interact with the nickel. So, already you have the sodium. So, this particular azide function is bridging your sodium and nickel. Is not that you are getting, so everything can be broken and you can have a bridging unit between 2 nickel sites, but it is not holding, is a very delicate molecule in that fashion. So, you are getting some chain and this particular chain is propagating. So, you have binuclear motif here, you have binuclear motif here and you are propagating that chain through nickel azide sodium and nickel azide sodium. Okay. Thank you.